Delaware Tech, Chemistry 100, Chemistry 110, Biochemicals. Biochemicals are the molecules that make up living organisms. So biochemistry is the study of the chemicals in these living systems, as well as the organization of the molecules in the cells and the various chemical interactions between them. We have four main categories of families of biomolecules, though this doesn't include every biomolecule, but most molecules within living systems fall into one of these four categories. Carbohydrates, which include simple sugars called monosaccharides, as well as sugars with two sugar units called disaccharides, and long polymers of sugars called polysaccharides. These are very polar molecules, and we'll talk about why that is. So they tend to be soluble in water, though the polysaccharides tend to not be as soluble. Lipids are the biochemist's name for fats. These are water insoluble. They tend to be very nonpolar because they're very rich in carbon and hydrogen, two atoms that form very nonpolar bonds. Molecules that are part of the lipids would include fatty acids, also known as free fatty acids, various oils that you might use in cooking that are derived from fatty acids, from plants, for instance, cholesterol, which I'm sure you've heard of. All your steroid hormones are lipids, so estrogen, testosterone, progesterone, those are all steroid hormones, as well as a lot of other hormones that are related to steroid hormones called corticosteroid hormones that are involved in a wide range of regulation of various metabolism processes in your body. Also bile salts. Bile salts and steroids are all derived from cholesterol. They have very similar structure. Bile salts are organic molecules that are somewhat soluble and somewhat insoluble, and therefore they act as detergents that your liver produces in order for you to, they're made in the liver and then they're transported to the intestines to mix with the fat that you eat in your diet in order to help solubilize the fat you've eaten so that you can digest it. Another category that's very important among the lipids are phospholipids. These are fatty molecules that have phosphate groups attached to one end of them. Phosphate groups are very polar, and then the lipid part is very nonpolar, so then they have two portions of them, and as a result they form very interesting structures. There's another molecule related to phospholipids called triacyl glycerides, sometimes just called triglycerides. That's why acyl is italicized here, and you may have heard of those. That is a storage form of fat that can float around your blood and go to cells, basically carrying fat to them to use potentially as energy. Proteins are composed of amino acids that are linked together in long chains, and then these chains can fold up into interesting shapes that have very specific functions. And there are many different kinds of proteins. If the protein is short, we call them a peptide. If they're long, we call it a polypeptide or a protein. So if it's only a couple amino acids, we call that a peptide. If it's more than 20 or 50, we might call it a protein. We have enzymes that are composed of proteins. Most of the enzymes in your body are predominantly made of proteins. There are some hormones in your body that are peptide hormones. There are also hormones in your body that are lipids. So please do not think that hormone means protein. But there are peptide hormones. The most famous one of those is insulin. There are proteins that are involved in transporting molecules across your membranes or within the cell. There are proteins that store things in your cells. Hemoglobin transports oxygen, but myoglobin stores oxygen. Those are two different proteins. Antibodies, the antibodies that fight disease in your body, are made of proteins. They're very specific Y-shaped proteins. Receptors, in other words, binding proteins that bind various hormones, any kind of hormone. These receptors are almost always made of protein, sometimes with carbohydrates on them, sometimes with lipids on them, but the bulk of them are made of proteins. So the receptors can be binding proteins, or there are other binding proteins that just bind things and turn on certain chemical reactions. We also have structural proteins, proteins that give the cell structure or things that give your body structure. We have proteins in your muscles called actin and myosin. Those are structural proteins. They're also contractile proteins. They're part of your muscles that contract and allow your muscles to flex. We also have proteins that are structural parts of your body, like collagen or keratin. That's part of your part of the component of hair. And then we have nucleic acids. The two major categories are DNA and RNA. DNA, the NA in both of these words stands for nucleic acid. And they carry genetic information. They carry the information that are your genes. They tell your body what to make, and usually in terms of what proteins to make. So they have the instructions for making proteins as well as the machinery to make the proteins, but not all of the machinery. There are actually other proteins as well as nucleic acids that make proteins. And then some of these RNAs are also involved in regulating genes. Proteins can also regulate genes, but now we know RNA can. So I know this is a very complex thing with a lot of overlap in function, but try to think of the generalities here. So let's talk about each of them. Carbohydrates. You can recognize carbohydrates because they are loaded with alcohol groups, OH groups on the carbons, and it makes them very polar. Here's glucose, and you can see there are 1, 2, 3, 4, 5 OHs on this particular version of glucose. There are two other versions of glucose that it switches back and forth between, and one other one also has 5 OHs. One only has 4 OHs, though. But the predominant form is to have 5 alcohol groups on it, 5 hydroxy groups on it. So this is very polar. It dissolves in water very well, as you probably know. So glucose, fructose, mannose, galactose, ribose, these are all single unit sugars, single carbon sugars. This one has a single ring, glucose. So you're going to look for single rings. That's a monosaccharide, one sugar unit. And you can tell sugars because they almost always end in OSE, fructose, mannose, galactose, glucose, allose. There are a lot of these that end in OSE. And they tend to be metabolites. We use them to burn for immediate energy. Disaccharides have 2 sugar units. They're attached to each other. Glucose attached to fructose in a very specific way is the sucrose that we eat as table sugar. Lactose, the sugar in milk, is galactose attached to glucose. Mannose are just 2 glucoses together, and each of these has very specific ways of linking them. This is way beyond the level of the course. I just want you to understand the examples and how you can recognize them by looking at a structure. So you're looking for these OHs all over on the carbons, and they tend to form rings very frequently. They are very useful as energy sources, either as immediate energy source or to storage as energy sources. The polymers of sugars are called polysaccharides. You link one after the other, and they can either be long chains or they can be, as shown in this picture, a branched chain. So we have that long chain at the bottom, and then we have 2 more sugars sticking out as a branch. Well, this is just a shortened version, but if you have these branches with literally hundreds or thousands of glucoses, single sugar units, linking them and coming off as branches, this is what the glycogen in your body, that's your animal starch, which is almost an identical structure to the plant starch that you eat. Plant starch comes in 2 varieties. One that is just straight chain with no branches, that's called amylose, but the one with branches shown here is called amylopectin, or amylopectin. And it's virtually the same structure as the starch you have in your body that you burn when you need energy after a while of doing exercise. So that's that's a couple different types of polysaccharides. Cellulose, which is one of the main components of wood and plant life. That's another polymer of glucose, but it's linked in different ways, so we can't break it down. Chitin, which is the main polymer that makes up the shells of shellfish, like crabs and shrimp. It's also one of the main components of the membranes of yeast. All of these are polysaccharides. So the polysaccharides can be used as energy storage, like the starches in the plants that we call starch, amylose and amylopectin, or the starch that we have in our body that we call glycogen. or they can be used as structural components like the cellulose or chitin. Chitin's also part of the insect exoskeletons as well. Lipids, how do you recognize lipids? Well, they're mostly made of carbon and hydrogen, and they tend to be very nonpolar. You find oxygens and nitrogens and phosphoruses and sulfurs, but they're not as common. Most of the molecule will be very nonpolar. So they tend to be hydrophobic, that is, they don't like being in water, they're not soluble in water, and they tend to be used for, the smaller ones tend to be used for energy. So we have free fatty acids. Remember, these are carboxylic acids, so there's a carboxylic acid at the end of these two that are on the left here, and a long chain of carbons. Sometimes they're double bonds, we call those unsaturated fatty acids, and sometimes there are no double bonds. They are loaded up with as many hydrogens as you can put on the carbons. They are saturated with hydrogens, and therefore those are saturated fatty acids, and that is where that term comes from. While it doesn't show it in here, the double bonds in unsaturated fatty acids actually form bends in the shape, and this is very useful in your body. So you want to have lots of these kinked or bent unsaturated fatty acids in the makeup of your cell membranes. We also have triglycerides that are made from 3 fatty acids attached to a glycerol. That's shown here in blue on the right. I skipped over that. We also have a different kind of lipid that's formed with 2 fatty acids attached to a different backbone. Instead of glycerol, it's attached to a molecule, it's attached to glycerol, but glycerol has a phosphate group on it that has something else. Now, I'm not going to go over the detail of this. You don't need to know it, but what you should know is that one part of the molecule where the phosphate is at is very polar and water soluble, and the other part is this long tail from the fatty acid that's very greasy and hydrophobic and nonpolar and doesn't want to be in water. And when you have a lot of these molecules together, the greasy long tail parts, what we call the hydrophobic tails, try to hide from the water, and the way they do that is they make a layer with the hydrophobic part in the center and the polar heads on the outside facing the water on either side of the membrane. And if you have enough of these, they make a sphere, and the sphere is made of this double layer, what's called a phospholipid bilayer. And this is how the membrane of every cell in your body is made, as well as the membranes of all the organelles in your cell. If you've taken biology, you'll know that organelles are smaller compartments in the cell. They all have membranes that are made of these phospholipid bilayers. Now, the phospholipid bilayers, if they have a lot of saturated fatty acids, they pack together so tightly they can barely move. You really want unsaturated fatty acids in there to make it harder for them to pack together so that things in the membrane, floating around in the membrane, like proteins, can move around freely. And if you don't get enough unsaturated fatty acids in your diet, your body can't make a lot of unsaturated fatty acids. Plants can make them, humans can't make that many. So what we make instead is cholesterol. And cholesterol gets in that membrane because it's also greasy and hydrophobic. It slips in the middle there and kind of breaks up the tight packing, making it more fluid. So cholesterol has a good reason to be in your body. You just don't want a lot of it, or you end up with obvious problems with heart disease and related syndromes. Steroid hormones are all made from cholesterol. So if you ever look at the formulas for the structures for estrogen and progesterone and testosterone, you'll see that they all have that same 4-ring structure of cholesterol. There are other kinds of hormones I'm not going to show here. It's way beyond the scope of the course, but there are also lipids called prostaglandins, which you probably have heard of, and leukotrienes, which you may not have heard of. Your fat-soluble vitamins. Being fat-soluble, they're all considered fats and fatty molecules and therefore called lipids. So anything that's not B or C is a fat-soluble vitamin. Waxes and oils are also lipids. They're very hydrophobic, they're nonpolar, they don't dissolve in water. The next category of molecules are the proteins. Proteins includes the polypeptides. Polypeptides really just another word for protein. The small versions of those are just called peptides. The longer versions, polypeptides. So these are made of monomers, monomers meaning single units that are linked together to form polymers, and each monomer is called an amino acid. Why are they called an amino acid? Well, because Every one of them has the same basic structure. You start with one carbon, that's the carbon in the middle of that structure, the one in black, and it has attached to it two functional groups, an amino group and a carboxylic acid group. So it is an amino acid. It has an amino group on that carbon and an acid group on the carbon, specifically a carboxylic acid. That leaves the carbon to have two more bonds. Most amino acids have just one hydrogen, and then the second position has some other group, and it can be something small, it can be something bigger. These groups are called side chains. We have 20 different naturally occurring amino acids that our proteins are made of. They all have a different side chain on them. So there are 20 different ones. The simplest one has just hydrogen in there as well, so it's a carbon with two hydrogens. That's called glycine, and then they get more complicated. You can always look at these when you're studying biochemistry. So these are linked together. They're called alpha amino acids because it's that first carbon where both the carbonyl and the amino acid, they're both attached to the same carbon, so it's called an alpha amino acid. You're not going to be tested on the alpha, but you should know that amino acids are linked together, and they're linked together by linking the amino group of one amino acid to the carboxylic acid group of another. And if you remember from your functional groups, so in circles here are each different amino acid residue, each part that was originally a separate amino acid, they are linked together with the carbonyl of one linked to the amine of another to form amide linkages. So if you look at this, there's a linkage here, a linkage here, a linkage here, a linkage here. Those are called amides. You already learned that from the functional groups. But in proteins, we call them peptide linkages. So peptide linkages are just a special kind of amide linkage between two adjacent amino acids. The R group, the side chain, gives the protein function. If you have all these different chemical groups sticking out from your long chain, you have all of these side chains together, it'll result in the protein folding together in some very specific pattern, and then the side chains that are sticking out from it can group together and have some very specific chemical role and give the molecule some kind of interesting function. So there are many different kinds of proteins with many different functions. We have enzymes like trypsin, which breaks down proteins, not all proteins, but some proteins. Amylase, which breaks down the amylose, the starch. Lipase, which breaks down certain lipids like the Acyl Glycerides and the phospholipids. DNA polymerase, which makes DNA. Kinases, which put phosphate groups on all kinds of molecules. But it's not important what these roles do. Just know that enzymes are catalysts. They speed up chemical reactions tremendously, and your body has literally a couple tens of thousands of enzymes in your body at any one time. Components of the immune system are also made of proteins. Antibodies, specifically the type of antibody called IgG, which stands for immunoglobulin G. Shown there in that picture, you see it's a Y-shaped molecule. And the top two parts of the Y are involved in binding foreign molecules. They'll actually stick together right here because they have a very specific shape. And your body at any one time has about 10 million antibodies in it. And so they can bind millions and millions of different molecules that you might be exposed to, especially from diseases. So that's very useful to have floating around in your system at any one time. Now, you're not going to be asked on the details. Just understand examples of them and basic functions of these and how to recognize the proteins. As I mentioned before, proteins can be involved in transport. Albumin is the most common protein in your blood. It's 60% of all the protein in your blood is albumin. Even though there are tens of thousands or even 10 million different proteins, depending on how you count them in your blood, one of those proteins accounts for 60% of all the protein in your blood. That is albumin. It's kind of the garbage scow of your blood system. It picks up and binds all kinds of things. It picks up and binds metals like calcium and some heavy metals occasionally. It binds drugs like aspirin. It binds sulfur compounds. It really is a versatile molecule that does a lot of different jobs in your body. It also binds fatty acids. Ferritin carries around iron, as the name ferra might suggest. Hemoglobin, as you know, carries oxygen. Storage molecules like ferritin can carry iron. Myoglobin can store oxygen. Albumin, as I mentioned, can store fatty acids. You have structural components like actin, myosin, keratin, collagen. And you have some hormones are made of protein, like insulin, glucagon. Glucagon is another important protein hormone. It does almost the opposite of what insulin does, so it's important to have both in your body. Growth hormone is a peptide hormone. It's made of protein. And oxytocin is a very short protein. I believe it's only 10 amino acids. And oxytocin has a role in many, many different biochemical and neurological pathways in your body. It's actually been associated with the bond between a mother and child when it's released while the woman is in labor. And then we have receptors. The receptors for almost every hormone are proteins. Insulin receptor, estrogen receptor, progesterone receptor, those are all made of protein. And the last category, nucleic acids. Deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. These are polymers of repeating monomers called nucleotides. So each repeating unit, this is the thing that's attached to another, to another, to another in long chains, is made of three things. It's made of let's start with the center. It's made with a sugar that has 5 carbons in it. And on one side of the sugar is a phosphate group. And on the other side of the sugar is what we call a nitrogenous base. That's a fancy word for saying a small molecule that looks aromatic. It's kind of aromatic and has amines in it. So it's made with nitrogens and carbons and hydrogens and maybe some oxygens. So it's an amine molecule that is attached to a carbon, and then the carbon also has a phosphate. And when they're linked, what you have is a backbone, and it's not shown here, so I'm going to show you. What you have is a backbone of sugar alternating with phosphate. So that's one of these groups, and then the sugar has some base sticking out. And there are 4 different kinds of bases for DNA, and there are 4 different bases for RNA. 3 of the bases are exactly the same, but the 4th base is different. It's thymine for DNA, what we just referred to as T, and it's uracil, U, for RNA. And that's how your body can tell the difference. If you sees T's in it, it knows it's DNA. If it sees U's in it, it knows it's RNA. Also, the sugar is slightly different between the two. So this is your monomer unit, and this is attached to another sugar and phosphate. Then that sugar has another base on it. And this is attached to another sugar and a phosphate, and that has another base. And this goes on and on. So this would be one strand, and these strands can be extremely long. And when I say extremely long, I mean millions of sugar phosphate sugar phosphates in that backbone structure with millions of bases attached to them in a very specific order. And that specific order of the bases, A, C, G, and T, is the order of your genetic code. So, and that code is found in the DNA as your primary source of the code, and then that's turned into pieces of RNA that mirror that code. It just replaces U for T's. And in DNA, you tend to have 2 of these strands together, so you'll have a second one over here where the sugar, and it goes in the opposite direction, so it doesn't really matter for this, but it'll go in the opposite direction, but it still looks like a sugar phosphate backbone, and every sugar has a base on it. And while you don't need to know this for the course, if one side base is an A, the other side has to be a T. If one side base is a C, the other side has to be a G. And these are linked together with 3 hydrogen bonds for GC pairs, and they're linked together with 2 hydrogen bonds for AT pairs. So they have hydrogen bonds that hold the strands in place. Now, I'm going to tell you a little secret here. 99% of all textbooks, probably closer to 100% of all textbooks, discuss the hydrogen bonds as holding the DNA together. And I'm going to tell you something, it's not required for the test, but it is a little piece of information that people should know more often that take biology and biochemistry, and that is that hydrogen bonds do not hold DNA together. In fact, they have almost no contribution to the total energy holding this double helix together. They're helix because they're spiral, and there are two of them wrapped around each other. The double helix, every textbook will tell you they're held together with hydrogen bonds. That's not true. The hydrogen bonds just tell the enzymes that are making DNA, oh, I see a A over here, so when I'm making this strand, I want to make sure there's a T over here. And when it sees the C over here, it makes sure it puts a G over here. And so you end up with this complementary pairing. A's are always matched with T's, C's are always matched with G's, always, 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 in DNA. And so while they hold it, they keep the bases somewhat stable next to each other, and they give specificity so that an A knows to be bound to a T and a C knows to be hydrogen bonded to a G. They do not contribute to the total energy of holding it together. That's actually something else that's not part of this course and is actually a bit complicated to describe. If you ever want to know, ask me. I'll be happy to explain it to you. But that was a little outside of the realm of the course. You should know that the DNA is double-stranded, the RNA is single-stranded. So let's look at the roles. DNA is your main source of your genetic code for most organisms. There are a few viruses that use RNA only or RNA in part as their genetic information, but almost every other living organism is DNA based. A lot of people don't count viruses as a living organism anyway, so we'll say that DNA is our genetic information. RNA is translated from the DNA in pieces, and that provides the information for coding for making protein. There are also types of RNA, and that RNA that gives the code for making protein is called messenger RNA or mRNA. We also have two other RNAs, tRNA and rRNA, and you're not responsible for the individual names here. Just know RNA and DNA. tRNA is transfer RNA and rRNA is ribosomal RNA. These RNAs are involved in actually making the protein. And then there's a whole category of many different types of small RNAs that are now known to be involved in regulating the genes, turning the genes on or turning the genes off. So RNA has many, many roles, and we didn't know about a lot of these roles as recently as 10 or 20 years ago. So the bases, as I mentioned, in DNA are A, C, G, and T, adenine, cytosine, guanine, and thymine. And in RNA, they're adenine, cytosine, guanine, and uracil, A, C, G, and U. A pairs with U in this case, but usually you get single-stranded RNA. It's not usually pairing with another strand. It might pair with it within the actual single strand, but that, again, is beyond the scope of the course. So I'll go over what you need to know for this. The sugars are different between the two. The sugar in RNA is ribose because it's ribonucleic acid, whereas the sugar in DNA is missing an oxygen, and therefore we call it 2-deoxy, missing an oxygen, ribose. Again, you don't need to know the details. The structure of DNA is usually double-stranded and linked with hydrogen bonds, but that's not really where the energy is to hold it together. And RNA is usually single-stranded, though it tends to form what are called hairpins and helices and stems and loops. So you might get RNA looping together and then suddenly forming this little thing called a stem and loop. So you might get some hydrogen bonds in here, making what we call a stem, and then you have a loop right here, and then this goes over here. Or this might end up forming some kind of spiral, a little helix on its own. So RNA can do a lot of things. Again, you're not going to be tested on that kind of detail. So what do you need to know? You need to know that it's made of nucleotides. You need to be able to recognize, if I show you a structure of DNA, look for that sugar that has a phosphate on one side and an amine base on the other. And if you see a bunch of these, especially if they're linked together, that's DNA. If it's only one strand, that's typically RNA. And that is it.